Right now, as you're listening to this, your liver is fighting a microscopic, multi-front war.
Yeah, a massive one.
I mean, it is actively neutralizing volatile compounds from the air you breathe, dismantling the microplastics that, slipped into your drinking water.
Right, and it's managing the metabolic exhaust produced by your own cells just to keep you alive.
Exactly. It's an organ of staggering, relentless biochemical engineering.
It really is.
But if you walk into literally any grocery store or just scroll through a social media feed, you are instantly bombarded by a completely different narrative.
Oh, detox narrative.
Yeah. You see shelves lined with like shrink-wrapped detox juices, herbal teas, 30-day cleanse kits.
And they're all making the same fundamental promise.
Right, that your liver is this dirty sponge, and if you just drink this $30 bottle of green liquid, you will miraculously flush out all the poisons.
Which is just not how biology works.
So today, we are wading directly into the hard science for this deep dive to find out why that narrative is not just a marketing fabrication, but a biological impossibility.
Yeah, because the clinical reality is actually far more compelling than the marketing.
Totally.
When we look at the stack of scientific literature on the table today, and we are talking robust human meta-analyses, double-blind, randomized controlled trials, granular nutritional reviews, we see a profound disconnect.
A huge one.
The public believes in a passive organ that needs to be scrubbed clean. But the data reveals a highly adaptive, proactive chemical laboratory.
Right, a lab that doesn't need to be cleansed, but rather requires very specific enzymatic substrates to execute its built-in waste management protocols.
Exactly.
So our mission today is to cut through that noise for you. We are going to examine how specific phytochemicals in foods, ancient spices, herbal extracts, how they actually interact with human biology.
And we need to establish the core premise immediately.
Which is?
No food, no juice, no supplement acts as a magical eraser for acute toxicity.
Right.
But they do operate as targeted biochemical catalysts for your internal defense systems.
I think we really have to dismantle that whole liver detox diet myth right at the foundation.
We do, because the phrasing itself is a clinical liability.
Wait, a liability? How so?
Well, if you are genuinely subjected to acute poisoning, say, heavy metal toxicity from industrial lead, arsenic exposure, cadmium, mercury.
Or like massive amounts of dioxins.
Right, or if you suffer a severe pharmaceutical overdose, a celery and apple smoothie is completely irrelevant to your survival.
Yeah, I mean, you're talking about situations that require immediate, aggressive pharmacology.
Exactly.
You can't juice your way out of severe heavy metal poisoning.
No, you need emergency medical intervention. You require intravenous chelating agents.
And those chemically bind to heavy metals in the bloodstream, right?
Yeah, so they can be excreted by the kidneys. Or you might need targeted antidotes like N-acetylcysteine for an acetaminophen overdose.
Which makes sense.
The wellness industry's co-opting of the word detox leads patients into incredibly dangerous territory.
Because they might delay actual life-saving medical treatment.
Exactly. They believe a dietary cleanse is a substitute for clinical toxicology.
So the phrase cleansing diet isn't just a misnomer.
No, it's a fundamental misunderstanding of human physiology.
Okay, so what's the correct framing then?
Based on the literature, it should be a diet supporting liver detoxification functions.
Supporting, not doing the detoxing for it.
Right. The liver is the engine actively performing the detoxification. It doesn't need to be clean.
It needs the raw materials required to sustain its own metabolic fires.
Yes, the compounds you ingest dictate whether that engine is operating with optimal catalytic efficiency or if it's just grinding its gears against a lack of chemical resources.
That makes so much more sense. Let's talk about where this data comes from, though.
Okay.
Because looking through these sources, we're not just looking at human trials.
Yeah, we're not.
A massive portion of the foundational nutritional research on hepatic function is conducted on animal models. We're seeing data on rats, quails, and even trout.
Which I know sounds strange.
Yeah. I mean, if I'm listening to this and I want to know how to manage my own health, it might seem disjointed to care about the liver enzymes of a fish.
It's a critical point regarding scientific methodology, really.
Yeah.
We utilize animal models to test extremes of toxicity that would be wildly unethical to test on humans.
Because you can't just intentionally poison a human study group.
Exactly. In these studies, researchers are intentionally exposing the subjects to lethal, catastrophic substances.
Right.
For instance, many of these trials utilize carbon tetrachloride.
Which is terrible stuff.
It is highly volatile. It's a synthetic chemical previously used as a dry cleaning solvent, and in fire extinguishers.
Wow.
And it is notoriously hepatotoxic. When metabolized, it unleashes a localized storm of free radicals that rapidly liquefy liver tissue.
Liquefy it. That's terrifying.
It is. Other studies use N-nitrosodiethylamine, which is a potent, aggressive carcinogen used to reliably induce liver cancer.
So you introduce a lethal industrial solvent to a rat to ensure the liver is pushed to the absolute brink of failure, and then you introduce the plant compound.
Exactly. We need to see if the botanical extract can intercept the devastation.
Got it.
If a specific vegetable extract or phytochemical can preserve the structural integrity of a rat's liver while it's being bombarded by a deadly industrial solvent.
Then it provides a very clear window into its cellular mechanisms.
Yes. It highlights the specific enzymatic pathways being upregulated.
And those pathways are the same in us.
Remarkably conserved across mammalian and even vertebrate evolutionary lines. We share those same baseline defensive architectures.
Okay, but that raises a broader question about our daily reality.
What's that?
If the liver is this incredibly evolved, self-cleaning biological laboratory, why do we need to artificially support it at all?
That's a fair question.
Like, if millions of years of evolution perfected this system, shouldn't we just get out of the way and let it function? I mean, think about a high-tech municipal waste treatment plant. A detox diet claims to be a magical wand that instantly vanishes the trash. But really, the foods we were talking about are like giving the plant workers better tools, a pay raise, and extra coffee so they can do their jobs more efficiently.
That's a great analogy.
But if the workers are already pros, why do they need so much extra support?
Because the environment that shaped that evolution no longer exists.
What do you mean?
The liver evolved to process naturally occurring stressors, right? A mild plant toxin, the occasional fungal spore on a piece of fruit.
Or the metabolic byproducts of, like, physical exertion.
Exactly. But the 21st century chemical landscape is entirely novel.
Oh, right.
We are constantly exposed to a relentless, unprecedented volume of synthetic chemicals.
Like microplastics shedding from packaging.
Yes, and agricultural pesticides, industrial PFAS or forever chemicals, and a massive influx of refined, ultra-processed dietary compounds.
So we are forcing an ancient biological system to process entirely foreign synthetics.
Yes, the sheer volume is the variable that changed.
And the metabolic burden is constant and overwhelming.
That is why supporting the organ's function is a physiological necessity, not a wellness fad.
Okay, so to understand how we actually do that, we have to look at the liver's primary method of dismantling threats.
Right, the two-phase detoxification system.
This is where the mechanics get genuinely fascinating. The liver doesn't just passively filter blood the way a kidney filters fluid.
Or the way a coffee filter catches grounds.
Yeah, it operates a sequential two-step chemical processing plant. Phase 1 and Phase 2.
So let's break down the biochemistry of Phase 1.
This is the modification phase, right?
Yes. The liver relies on a massive family of enzymes, primarily the cytochrome P450 superfamily.
That sounds like a sci-fi robot.
And it kind of acts like one. These enzymes are the first line of defense against lipid-soluble toxins.
So what's their main job?
Their job is to physically alter the chemical structure of the toxic molecule, usually by adding an oxygen atom.
Oh, to change how it behaves?
Right. They are trying to make the toxin more hydrophilic or water-soluble so it can eventually be flushed out.
Now, the sources specifically cite grapefruit juice in relation to this Phase 1 activity?
Yes, they do. In several controlled animal studies, administering grapefruit juice significantly modulated the activity of specific cytochrome P450 enzymes.
Wait, really? Just normal grapefruit juice?
Yes. Researchers exposed subjects to 12-well-2-dimethylhydrazine, which is a highly potent colon cancer-causing agent.
Wow.
And the compounds in grapefruit juice upregulated the liver's ability to initiate the breakdown of that specific carcinogen.
That's incredible. But Phase 1 has a very dangerous catch, doesn't it?
It does.
Because when you run a chemical through the cytochrome P450 system and modify its structure, you sometimes inadvertently create a compound that is temporarily more volatile.
And more dangerous than the original toxin.
Yeah.
That is the paradox of Phase 1. The intermediate metabolites it produces are highly reactive.
They're missing electrons, making them free radicals.
Exactly. And if they aren't immediately neutralized, they will violently steal electrons from your healthy cell membranes.
And proteins and DNA.
Right, causing massive oxidative stress and tissue damage.
Think of Phase 1 like a bomb disposal unit, right? They decide that the best way to deal with a dormant bomb is to strip off its thick metal casing.
Good analogy.
Yes, you have exposed the wiring so you can defuse it, But for a brief window, you have a highly sensitive, exposed explosive sitting in the middle of the room.
Very sensitive.
If the second team isn't standing by to immediately cut the wire and neutralize it, the bomb goes off.
And the second team is phase 2.
The conjugation phase?
Yes. Phase 2 enzymes take that highly reactive, stripped-down intermediate metabolite from phase 1, and they chemically bind or conjugate it to another, safer molecule.
They pack it in a safe box, basically.
Right. They might attach a glutathione molecule or a sulfate group or an amino acid.
And this binding process immediately neutralizes the volatility.
Exactly. And renders the entire package highly water-soluble.
At that point, it can be safely excreted into the bile to leave via the intestines.
Or into the blood to be filtered by the kidneys.
And this intricate handoff between phase 1 and phase 2 is exactly where our dietary choices intervene.
It is. The data points heavily toward cruciferous vegetables.
Like broccoli, Brussels sprouts, kale. We hear about them constantly as superfoods.
But there is an actual mechanism inside the liver.
What is that mechanism?
It relies on specific phytochemicals called glucosinolates and isothiocyanates.
Okay, big words.
Yeah, but the most heavily researched one is sulforaphane, which is found abundantly in broccoli sprouts.
Oh, I've heard of sulforaphane.
It's a profound metabolic modulator. It enters the cell and activates a specific protein pathway called NRF2.
NRF2.
Yes. You can think of NRF2 as the master switch for the body's antioxidant response.
Wow.
When sulforaphane flips that switch, it simultaneously upregulates both phase 1 and phase 2 enzymes.
So it ensures that the bomb disposal unit and the neutralization team are perfectly synchronized. You don't have phase 1 stripping casings faster than phase 2 can diffuse them.
It optimizes the entire assembly line, preventing a backlog of toxic reactive intermediates from destroying the hepatic tissue.
That's amazing. And we see similar targeted support from other dietary sources that are incredibly popular right now, like kombucha.
Right, kombucha.
Kombucha is ubiquitous. It's pitched as a cure-all gut health beverage.
It is.
But we are looking at its specific application to the liver. Is there an active mechanism there, or is it just the placebo effect of drinking expensive fermented tea?
The clinical literature actually isolates a very specific, highly functional compound generated during the fermentation of kombucha.
It is.
Glucuronic acid.
Yeah.
This compound is critical for a specific type of phase 2 conjugation called, appropriately, glucuronidation.
So glucuronic acid actively binds to toxic metabolites.
And xenobiotics, which are simply any chemical substances foreign to the biological system.
So the glucuronic acid from the kombucha acts as the direct neutralizing agent in phase 2. It's the chemical cap placed on the exposed explosive.
Exactly, it binds to the foreign substance, rendering it inert and ready for excretion.
Wow.
And the studies highlighted in the sources show kombucha demonstrating remarkable protective effects against severe biological stressors.
Including paracetamol toxicity, right?
Yes. Paracetamol, widely known as acetaminophen, is metabolized by the liver, and in high doses, it depletes the liver's natural antioxidant stores.
Leading to acute failure.
Right. But glucuronic acid provides an alternative conjugation pathway, sparing the liver's internal resources.
And it also demonstrated mitigation against our old benchmark toxin, carbon tetrachloride.
And even systemic lead exposure.
Which is wild for a fermented tea. And green tea operates in a similar defensive capacity, doesn't it? But utilizing different compounds.
Right. Green tea relies on its dense concentration of polyphenols, specifically catechins like EGCG.
EGCG?
The data indicates these catechins operate both as direct free radical scavengers and as modulators of the detox enzymes.
So they physically intercept the oxidative damage caused by things like agricultural pesticides.
And industrial polychlorinated biphenyls, or PCBs, heavy metals like arsenic, and mycotoxins produced by molds.
The idea that the catechins in a cup of green tea are actively floating through your hepatic circulation, literally physically intercepting the oxidative blast radius of an agricultural pesticide, is staggering.
It really demonstrates the profound biochemical interface between human physiology and the plant compounds we ingest.
Absolutely.
These green defenders, the cruciferous vegetables, the fermented glucuronic acid, the tea catechins, they are exceptional for managing the daily systemic load of environmental toxins.
They keep the phase 1 and phase 2 pathways functioning efficiently.
Yes, but environmental toxins are only half the battle.
Right, because we have to address what happens when the liver is compromised not by a foreign chemical, but by the relentless accumulation of metabolic fuel.
Which brings us to a massive global issue.
Yes, we are pivoting to NFLD, non-alcoholic fatty liver disease.
It's a huge problem.
When most people think of severe liver pathology, they immediately associate it with chronic alcohol abuse.
Right, cirrhosis driven by liquor.
But NFLD is a completely different mechanism, and it is arguably the most pervasive chronic liver illness on the planet right now.
It is a global epidemic, inextricably linked to metabolic syndrome, insulin resistance, and modern dietary patterns.
It's lipid accumulation, right?
Yeah.
Fat physically displacing healthy liver tissue.
Yes, in patients who consume little to no alcohol, the sheer volume of fat causes cellular swelling, chronic inflammation, and eventually fibrotic scarring.
But wait, if someone has fatty liver, the obvious answer seems to be stop eating fat.
You would think so.
Right, because the Mediterranean diet is loaded with fat. Olive oil, fatty fish, nuts.
Yes, it is.
If a patient's organ is already choking on trapped lipids, why are we pouring more dietary fat into the system? That sounds like a fast track to advanced disease.
I get why that sounds counterintuitive,
Yeah,
but that assumes all lipids behave identically within our metabolic pathway.
which they don't.
No, which is a fundamental misunderstanding of how NAFL develops. The disease is rarely caused by the consumption of high-quality monounsaturated fats or omega-3 fatty acids.
Okay, so what is the primary driver?
The primary driver of NAFLD is the overconsumption of refined carbohydrates, fructose, and sugars, combined with systemic insulin resistance.
Sugar.
Yes. When you flood the liver with excess glucose and fructose, it initiates a process called de novo lipogenesis.
De novo lipogenesis, meaning making new fat.
Exactly. The liver literally synthesizes brand new fat molecules out of the sugar.
Wow.
And because the system is overwhelmed and insulin resistant, it cannot export those fats. They become trapped in the hepatocytes.
The liver cells. So a diet heavy in processed cereals and sugary beverages is the true culprit behind a fatty liver, not an avocado or a handful of almonds.
Precisely. The fats profile of the Mediterranean diet, specifically the oleic acid in olive oil and the EPA and DHA in fish, actually improves cellular insulin sensitivity.
So they help the situation.
Yes, they downregulate inflammatory cytokines within the liver, and they facilitate the lipid oxidation pathways, helping the liver burn and export the trapped fat.
And we have robust human data to validate this, right? Specifically, an intensive randomized controlled trial highlighted in our sources.
Yes, a very well-designed trial.
Let's look at the methodology of this trial. They took 180 subjects diagnosed with NAFLD, and they monitored them over a 12-week period.
Right.
They divided them to test the efficacy of the Mediterranean diet, but they added another critical variable.
Beetroot juice.
Yes, specifically a 250 milligram daily dose.
So they established a control group, a group adhering strictly to the Mediterranean diet, a group receiving only the beetroot juice supplement, and a combination group receiving both interventions.
And the clinical markers they tracked provide a comprehensive map of hepatic function.
They do.
The results across the board were profound. Let's walk through these specific clinical markers because anyone who has ever looked at a comprehensive metabolic blood panel has seen these acronyms, often without any context.
Yeah, can be confusing.
The trial showed significant reductions in bilirubin and alkaline phosphatase, or ALP. What are those telling us?
Well, bilirubin is a yellowish pigment that is produced during the normal breakdown of red blood cells.
Okay.
A healthy liver continually processes bilirubin and excretes it into the bile.
So, if it's high...
If bilirubin levels are elevated in the blood, it indicates the liver's processing capacity is structurally compromised.
This is what causes jaundice, right?
Exactly. And alkaline phosphatase, or ALP, is an enzyme related to the biliary ducts.
Okay.
High ALP often indicates that the bile flow out of the liver is blocked or severely inflamed.
So the fact that the beetroot and Mediterranean diet combination lowered both of these signifies that the internal processing and the exit pathways were clearing up.
Yes, it means the pipes are unclogging.
But the most crucial markers, the ones that really signal acute ongoing damage, are ALT and AST.
Right.
And the trial showed massive reductions in these two enzymes. Why does the listener need to know about ALT and AST?
Because alanine transaminase, or ALT, and aspartate transaminase, AST, are the primary indicators of cellular distress.
Where do they come from?
These enzymes belong inside the liver cells, the hepatocytes. They're required for amino acid metabolism.
Inside the cell, so they shouldn't be in the blood.
Right. In a healthy organ, they remain enclosed within the cell membrane. But when a hepatocyte becomes engorged with fat, when it suffers oxidative stress and the lipid bilayer begins to break down, the cell membrane becomes permeable.
The structural integrity of the cell fails, and the contents spill out.
Exactly.
If the cell is a water balloon, the swelling and inflammation stretch the rubber until it develops microscopic tears, and the water inside leaks into the surrounding environment.
That is exactly what happens. The ALT and AST leak out of the dying hepatocytes and circulate in the bloodstream.
So an elevated ALT score on a blood test is quite literally a measurement of how many liver cells are actively rupturing.
Yes, it's a distress signal.
But over 12 weeks, the beetroot juice and Mediterranean diet protocol drastically suppressed those numbers.
It did. The inflammation subsided, the cell membrane stabilized, and the leaking stopped.
That's incredible.
Most importantly, the intervention physically reduced hepatic steatosis.
Meaning the actual volume of fat trapped within the tissue.
Yes.
Now, as impressive as the blood markers are, the trial examining artichoke leaf extract, or ALE, provided physical, visual evidence of this reversal that is hard to ignore.
Yeah, the ALE study was another rigorous double-blind RCT involving 100 subjects with ultrasound-confirmed NFLD.
And what did they do?
They administered 600 milligrams of artichoke leaf extract daily for 2 months.
Okay.
Blood markers like AST and ALT improved, but researchers utilized Doppler ultrasound to map the actual hemodynamic.
The physical movement of blood.
Yes, through the liver architecture.
So I compare the artichoke extract to a master plumber, right?
Well, that's a good way to look at it.
Because it's not just tweaking chemicals, it's literally opening up the pipes.
Right, it is modulating endothelial function. Think of the portal vein as the massive central highway delivering nutrient-rich blood from the digestive tract directly into the liver.
Okay.
In a patient with NAFLD, the liver is swollen with fat and choked by fibrotic tissue.
So it's a tight squeeze.
It creates immense physical resistance. The blood struggles to enter, causing portal hypertension.
The portal vein physically distends and dilates because of the back pressure.
Yes, exactly.
It's a traffic jam causing the main highway to swell.
But after 2 months of artichoke leaf extract, the Doppler ultrasound revealed a significant reduction in the diameter of the portal vein.
This swelling subsided.
Furthermore, it showed increased blood flow velocity through the hepatic vein, which is the exit route.
So the pressure within the organ dropped, the physical resistance vanished, and healthy circulation was restored.
It essentially shrank the swollen liver back to its optimal dimensions.
Wow. It also improved their APRI score, right?
Yes, the AST to platelet ratio index.
What does that measure?
It is a highly reliable, non-invasive metric used to estimate the severity of liver fibrosis, or scarring.
Okay.
As liver damage progresses, the body's platelet count often drops while EST rises.
Oh, so a lowering APRI score strongly suggests that the artichoke extract wasn't just clearing the fat, but halting the actual structural scarring process of the organ.
It's remarkably powerful.
We've seen how beetroots and artichokes handle dietary fat and metabolic stress.
Yeah.
But what about extreme external toxins and heavy metals? Because an AFLD is a slow burn. Heavy metals are an aggressive structural assault.
Very true.
So for that, we turn to the heavy metal shields. We touched on lead and arsenic, but the clinical mechanisms behind how plants shield against these metals are wild.
They are.
Let's go back to beetroot. It isn't just for metabolic fat.
No, the literature emphasizes that beetroot possesses targeted protective mechanisms against lead poisoning.
Really?
It demonstrably reduces the accumulation of heavy metals not just in the blood, but sequestered within the liver tissue itself.
Okay, wait. How does a root vegetable physically alter the absorption and retention of a toxic heavy metal like lead?
It hinges on a class of highly bioactive pigments called betalains.
Betalains.
These are the compounds responsible for the intense red coloration of the beet.
Oh, interesting.
To understand their function, we have to look closely at what heavy metals actually do at the cellular level. When lead, or cadmium, infiltrates hepatic tissue, it operates as a relentless generator of oxidative stress.
We keep coming back to oxidative stress. It is the underlying mechanism of almost all this damage.
Because it is the fundamental mechanism of cellular degradation. Heavy metals induce the overproduction of reactive oxygen species, or ROS.
And these are highly unstable molecules violently seeking electrons.
Yes, they will rip electrons away from the lipid membranes of the liver cells.
A process called lipid peroxidation.
Right, it initiates a chain reaction of cellular destruction. The betalains in beetroot intervene directly in this process.
How so?
They are powerful electron donors. They flood the tissue and voluntarily surrender their electrons to the reactive oxygen species.
Neutralizing them before they can tear apart the cell membranes.
Exactly. By quenching that fire, the betalains preserve the cellular structure, preventing the lead from causing irreversible necrotic damage.
So the beetroot is basically providing the chemical fire retardant to stop the oxidative damage.
That's a great way to picture it.
But the sources highlight another compound that takes a different approach to heavy metals, chlorella.
Ooh, yes.
This is the bright green microalgae that has completely saturated the supplement market.
And the clinical data on chlorella is surprisingly robust,
Yeah.
particularly regarding its capacity for chelation.
Chelation.
Yes. We are looking at a comprehensive meta-analysis encompassing 7 different randomized controlled trials.
Wow, 7.
The aggregated data confirms that chlorella supplementation significantly suppresses AST levels, showing a clear hepatoprotective effect, especially in patients already burdened by NAFD.
But its defining characteristic is its ability to accelerate the clearance of heavy metals like lead and cadmium.
And highly toxic synthetic dioxins.
But wait, chelation usually involves intravenous drugs in a hospital. How is an oral algae supplement accelerating the removal of cadmium?
It is a process of physical and chemical binding within the gastrointestinal tract and the biliary system.
Like a chemical sponge.
Exactly.
Mm-hmm.
The cellular architecture of chlorella is unique. Its cell wall is composed of complex mucopolysaccharides and specific glycoproteins that have a high binding affinity for heavy metal ions.
Okay.
As the liver filters out these toxins and excretes them into the bile empties into the intestines.
But the intestines often just reabsorb the toxins and send them right back to the liver, right?
Exactly. It's called enterohepatic circulation.
Ah.
It's a vicious cycle where the liver keeps trying to throw the trash out and the gut keeps bringing it back inside.
That sounds exhausting.
It is. But the mucopolysaccharides in the chlorella intercept this cycle. They physically bind to the heavy metals in the gut, trapping them in an indigestible matrix.
So they are permanently excreted from the body rather than reabsorbed.
Yes. However, the meta-analysis highlighted a critical physical caveat regarding how chlorella is manufactured.
Right. The efficacy is entirely dependent on the physical processing of the supplement, the broken cell wall.
Exactly. The cell wall of chlorella is exceptionally thick and indigestible by human enzymes.
So if you just eat it whole.
If you consume intact chlorella, the heavy metal binding compounds remain locked inside the cell, completely unavailable to your digestive tract.
Useless.
Basically. The clinical benefits, specifically the lowering of ALT enzymes and the chelation of metals, were heavily dependent on the supplement utilizing a broken cell wall or cracked cell wall manufacturing process.
The physical structure of the algae must be pulverized to expose the active binding sites.
Right. If the cell wall isn't broken, the chlorella just passes through your system completely inert.
Now, there is another heavy metal interceptor in the sources that approaches this from yet another angle, and it involves one of the strangest study designs I've ever seen.
The Trout study.
Yes, coriander or cilantro. I have to ask, why are scientists feeding coriander to fish? How does a trout's liver help you or me understand our own bodies?
I know it sounds inherently absurd to extrapolate human liver health from the dietary habits of a fish.
It kinda does.
But in the realm of environmental toxicology, aquatic models are foundational. Fish are constantly submerged in the chemical runoff of human industry.
Oh, that makes sense.
They are the ultimate bioindicators of heavy metal accumulation in tissue.
Okay, so in this study, the researchers supplemented the diet of these trout with coriander and then intentionally exposed them to cadmium.
Which is a highly toxic, carcinogenic heavy metal used in battery manufacturing and industrial plating.
Right, and what happened?
The physiological outcome was striking. The trout that received the coriander supplementation accumulated 20 to 25% less cadmium within their hepatic tissue compared to the control group.
Wow. Reducing heavy metal accumulation by a quarter simply through the introduction of a common culinary herb.
Yes.
How is the coriander stopping the cadmium from embedding in the liver?
Well, while chlorella relies on the physical sponge of its cell wall in the gut, coriander utilizes a different array of phytochemicals, likely involving plant-based chelators called phytokeratins.
Phytokeratins.
These compounds interact with the metal ions, either blocking their transport mechanisms across the intestinal lumen or actively binding them within the bloodstream to facilitate rapid renal excretion.
Oh, so before the liver is forced to sequester them.
Exactly. Other trials confirm its efficacy extends beyond metals. Coriander demonstrated potent protection against our benchmarks of chemical devastation, thiacetamide, and carbon tetrachloride.
It's incredible that simple plants can bind to heavy metals. But nature's pharmacy goes even deeper.
Much deeper.
Some of our most potent, scientifically validated liver protectors have been sitting in our spice cabinets and apothecary jars for thousands of years.
Yes, and modern clinical trials are now validating them at the molecular level.
Let's start with garlic.
Garlic is the quintessential intersection of culinary tradition and potent pharmacology.
What does the data say?
The animal data sets a clear baseline for its efficacy. We look at studies involving aquatic models exposed to aflatoxins.
Aflatoxins, right.
Aflatoxin B1 is a profoundly dangerous mycotoxin produced by Aspergillus molds, which frequently contaminate agricultural staples like corn and peanuts.
Yikes.
It is one of the most hepatotoxic and aggressively carcinogenic substances found in nature.
So when the subjects were exposed to the aflatoxin, what was the physiological marker of the damage and how did the garlic intervene?
The primary biomarker researchers measured was melandialdehyde.
Melandialdehyde, okay.
When the aflatoxin initiates that devastating chain reaction of lipid peroxidation, where free radicals shred the fat molecules in the cell membranes, the chemical byproduct left behind in the wreckage is melandialdehyde.
Oh, so spiking levels of this compound indicate that the liver cells are actively rusting and disintegrating.
Exactly. And the administration of garlic profoundly suppressed the melandialdehyde levels.
It arrested the lipid peroxidation.
Yes.
And we have the human trials to back this up, right? Not just fish data.
Right. We have an exceptionally strong meta-analysis comprising 4 distinct human randomized controlled trials, observing 212 patients suffering from chronic liver disease.
And the results?
The clinical data is unequivocal. Consistent garlic supplementation yielded statistically significant reductions in both AST and ALT enzymes.
That's huge.
It didn't stop there. It improved their systemic lipid profiles, lowering total cholesterol and LDL, and actively facilitated an overall reduction in body weight.
So it is a multi-pathway metabolic regulator.
It really is.
But when we discuss herbal regulators, there is an undisputed heavyweight champion of liver health.
Oh, absolutely.
A plant with a 2,000-year documented history of medicinal use. Milk thistle, or Silybum marianum.
The clinical history of milk thistle is captivating. Ancient physicians, completely devoid of an understanding of cellular biology, utilized milk thistle extracts for snake bites, and crucially, for acute mushroom poisoning.
Specifically, poisoning from the death cap mushroom, Amanita phalloides.
Yes. The death cap contains a class of toxins called amatoxins. When ingested, amatoxins are rapidly absorbed by the liver.
Where they aggressively inhibit the enzyme RNA polymerase II.
Right. And without this enzyme, the liver cells cannot synthesize proteins, and the cells undergo rapid, catastrophic necrosis.
The organ simply liquefies over a matter of days.
It's horrific. Historically, intravenous silibinin, the primary active component in milk thistle, was the only intervention that provided victims a statistical chance of survival.
Wait, if it's protecting against a mushroom toxin that stops protein synthesis and liquefies the organ, how is the milk thistle physically stopping the destruction? Is it neutralizing the toxin in the bloodstream before it hits the liver?
Actually, it operates directly at the cellular membrane. Silibinin acts as a competitive inhibitor.
Meaning?
Think of the surface of the liver cell as having very specific locks, and the amatoxin is a key, perfectly designed to fit that lock, open the door, and destroy the interior.
Oh, okay.
Silibinin alters the physical structure of the hepatocyte's outer membrane. It effectively changes the locks on the doors.
So the amatoxin can no longer bind to the receptors?
And it is blocked from penetrating the cell interior. It is forced to remain in the bloodstream, where the kidneys eventually clear it.
So it physically barricades the cell, like a cellular riot shield that historically stood between a deadly mushroom toxin and the liver's cells.
It does. And beyond that immediate defensive barrier, silymarin, the broader complex found in milk thistle, is deeply regenerative.
Regenerative how?
It actively stimulates ribosomal RNA synthesis within the surviving liver cells, accelerating their ability to divide, repair, and replace the destroyed tissue.
Wow.
It protects against acetaminophen toxicity and phenylhydrazine exposure. It is both a shield and a regenerative catalyst.
Now, while milk thistle focuses on the cellular membrane, turmeric, or specifically its active phytochemical, curcumin operates deep within the internal machinery.
It does.
We always hear about curcumin for joint inflammation, but its role in hepatic function is equally profound.
Curcumin is a highly potent upregulator of the phase 2 detoxification pathways we discussed earlier.
The conjugation phase.
Yes, it enters the hepatic environment and signals for the massive overproduction of specific phase 2 enzymes, particularly glutathione S-transferase and quinone reductase.
Okay, so it ensures the liver has a massive surplus of the binding agents required to neutralize reactive metabolites from alcohol, toxicity, paracetamol overdoses, and heavy metal exposure.
Exactly. But the clinical application of turmeric comes with a massive physiological obstacle.
Right. Okay, we've talked about garlic, turmeric, cloves, and black cumin. Should the listener just start swallowing spoonfuls of their spice rack every morning? What does the science say about bioavailability, like with turmeric?
You're right to bring that up. If you simply ingest a tablespoon of raw turmeric powder, your liver might never process a single molecule of curcumin.
Really? None.
Curcumin possesses abysmal pharmacokinetic bioavailability.
Yeah.
The moment it enters the gastrointestinal tract and the liver, it undergoes rapid glucuronidation.
Which is, wait, the exact phase 2 process we want it to enhance?
Ironically, yes. That process paradoxically tags the curcumin itself for immediate destruction and excretion.
Oh, so it is metabolized and eliminated before it can exert any systemic effect.
Yes, it needs a pharmacological chaperone to survive the digestive process.
It requires piperine, the active alkaloid compound found in black pepper.
Exactly. Piperine acts as a targeted inhibitor of the specific digestive enzymes that destroy curcumin. By temporarily shutting down that specific pathway in the gut and liver, piperine allows the curcumin to pass through into the systemic circulation intact.
So clinical trials demonstrate that the co-administration of curcumin with piperine enhances the bioavailability.
By an astonishing 2,000%.
2,000%, wow.
Yes, without the piperine, the curcumin is destroyed at the border.
The turmeric is like a general who needs black pepper as his armored transport vehicle to actually safely escort the curcumin directly to the hepatic battlefield.
It is the difference between an inert culinary flavor and a potent botanical medicine.
That's wild. And speaking of culinary flavors, the sources shed light on another common spice with surprising clinical utility: cloves.
Yes, cloves.
We usually relegate cloves to holiday baking, but the chemistry is aggressive.
Very. Cloves contain a highly bioactive phenolic compound called eugenol.
Eugenol.
The data indicates eugenol operates as a potent neutralizer of environmental xenobiotics.
Like what?
It provides substantial cellular protection against alcohol-induced hepatotoxicity, mitigating the oxidative damage caused by ethanol metabolism.
Oh, wow.
There is also emerging compelling clinical evidence suggesting that eugenol may actively alleviate the physiological symptoms associated with cirrhosis, the late-stage fibrotic scarring of the organ.
From a baking spice. That's incredible. But we have to cover the final ancient protector highlighted in the clinical data: black cumin seed, or Nigella sativa.
Right.
The methodology behind the trial studying this seed is incredibly thorough.
It is a meticulously designed study utilizing an avian model,
Right.
specifically 432 quails monitored continuously over a 35-day period.
Quails.
Yes, avian models, particularly quails, are frequently utilized in metabolic research because they possess rapid metabolic rates and specific lipid storage mechanisms that closely mirror human pathogenic states under dietary stress.
So they divided these 432 quails into distinct groups to measure the dose-dependent response to black cumin seed.
They supplemented the baseline feed with strictly controlled variables: 0.5%, 1%, and 2% black cumin seed integration.
And what were they looking for?
They were analyzing the comprehensive biochemical landscape of the liver. The data returned clear, dose-dependent efficacy.
Meaning, the more seed, the better the result.
Yes, the supplementation suppressed AST levels, indicating preserved cellular integrity. It drove down lactate dehydrogenase, or LDH, an enzyme that spikes when tissues are deprived of oxygen and forced into anaerobic glycolysis due to structural damage.
Wow.
It lowered systemic urea and circulating LDL cholesterol. But the most critical revelation was its interaction with glutathione.
Glutathione is arguably the most vital molecule we've discussed today. It is the body's master endogenous antioxidant.
It is the primary currency the liver uses to pay off oxidative debt.
Right, so every time a heavy metal, a synthetic chemical, or a volatile metabolite threatens a cell, the liver spends a molecule of glutathione to neutralize it.
Exactly. If the liver depletes its glutathione reserves, the organ rapidly succumbs to necrotic failure.
And what did the black cumin do?
The clinical data proved that the black cumin seed actively stimulated the liver's internal synthesis of glutathione.
It forced the organ to stockpile its most valuable defensive weapon.
Yes. And the physical outcome for the birds receiving the highest doses of the black cumin was totally unexpected.
What was it?
The quails receiving the 1% and 2% supplementation demonstrated a statistically significant reduction in the spontaneous development of fatty liver syndrome.
So the black cumin wasn't merely reacting to an injected toxin.
No, it was actively optimizing the underlying metabolic lipid pathways, preventing the onset of disease. It maintained the critical balance between oxidant stressors and antioxidant defenses.
Man, we have covered a massive expanse of clinical terrain.
We really have.
We've analyzed the hemodynamics of artichoke extract, the mucopolysaccharides of chlorella, the betalains in beetroot, and the complex phytochemicals of garlic, turmeric, cloves, and black cumin.
It's a lot of data.
We've seen how they regulate cytochromes, block amatoxins, and chelate heavy metals. But when you step back and look at this vast, disparate collection of plants and spices, an overarching question emerges.
What's that?
Why? I mean, why do a root vegetable, a marine algae, and an ancient seed all possess these highly specific, perfectly calibrated chemical keys that unlock human liver enzymes?
Ah, right. The answer lies in the harsh realities of evolutionary biology and a concept known as xenohormesis. Plants do not synthesize these incredibly complex, metabolically expensive phytochemicals for human benefit.
They synthesize them for their own survival.
Exactly, because a plant cannot fight or flee. It is anchored to the soil.
It is entirely exposed.
It must endure scorching UV radiation, systemic drought, aggressive fungal pathogens, the exact same aflatoxin molds we discussed, grazing predators, and toxic heavy metals in the soil.
So to survive that gauntlet over millions of years, plants evolved intricate, aggressive biochemical defense systems.
They manufactured silymarin, curcumin, and eugenol to protect their own cellular structures from oxidative collapse.
And because humans and our vertebrate ancestors evolved in constant contact with those plants consuming them for millions of years, our physiology adapted.
We learned to recognize those plant stress compounds.
We effectively hijacked the botanical defense network.
Exactly. When we ingest these phytochemicals, our internal sensors, like the NRF2 pathway, recognize them as mild cellular stressors.
Oh, they aren't enough to cause damage, but they are enough to trigger an alarm.
Right. That alarm commands the liver to radically upregulate its own endogenous defenses, to synthesize more glutathione, to activate phase 2 enzymes, to fortify the cell membranes.
We are borrowing the chemical resilience forged by plants over millions of years of evolutionary warfare.
It reframes our entire relationship with nutrition, really.
We aren't just extracting caloric energy from a plant. We are downloading survival data from the environment directly into our cellular machinery.
That is the functional takeaway from all this clinical data. You cannot reverse acute industrial poisoning with a juice cleanse.
Right.
But you can intelligently integrate these validated botanical compounds into your daily routine.
If you understand that combining curcumin with piperine alters your phase 2 pathways.
Or if you recognize that broken cell wall chlorella intercepts heavy metals in the gut.
Or that beetroot and artichoke extracts can mechanically alleviate the portal hypertension caused by metabolic syndrome.
Then you are no longer a passive victim of a toxic environment. You are actively engineering your physiological resilience.
So we started this journey navigating the murky, often deceptive waters of internet detox trends. We discarded the myth of the overnight miracle cleanse.
Because you cannot simply sponge away the metabolic burden of modern life.
Exactly. But in replacing that myth with biochemistry, we uncovered something far more powerful. An organ of staggering adaptability. We saw how it can literally rebuild its own cellular architecture, clear blockages, and neutralize the most aggressive toxins on Earth, provided it is given the correct phytochemical substrates.
It possesses the most profound regenerative capacity of any organ in the human body. But that capacity requires specific environmental inputs to function.
It is a remarkable system.
Okay.
But as we close this deep dive, I want to leave you with one final provocative consideration regarding the future of that system.
Okay.
We have spent this time detailing how the human liver evolved over millions of years to decipher and utilize ancient plant compounds. But our current chemical environment is changing at a pace that evolution cannot match.
It really is.
Every single year, human industry synthesizes and releases thousands of entirely novel forever chemicals.
Complex polymers, PFAS, and microplastics.
That quite literally did not exist on this planet a century ago. We are flooding our internal architecture with alien molecules.
We are presenting the cytochrome P450 enzymes with a structural puzzle they have never encountered in the history of our species.
But consider the vastness of the natural world we haven't yet cataloged.
Okay.
If our livers can hijack the compounds of a common thistle or an everyday spice to survive lethal threats, What else is out there?
That's a fascinating question.
Is it possible that deep within the unstudied biodiversity of the Amazon basin, or hidden inside an undocumented strain of deep ocean algae, there exists a unique phytochemical architecture?
A new biological key.
Exactly. Could our livers, if exposed to a plant we haven't even discovered yet, learn to upregulate entirely new enzymatic pathways capable of dismantling the synthetic forever chemicals we currently believe are indestructible?
I mean, the ultimate antidote to the industrial age might not be synthesized in a pharmaceutical lab.
It might be a silent ancient defender currently growing, undiscovered in the wild.
The biochemical ingenuity of the natural world has consistently outpaced our own. It is highly probable the chemical solutions already exist. We simply have to find them.
It fundamentally changes how you view that complex, constantly adapting laboratory inside you. It isn't a passive filter waiting to fail. It is a dynamic ecosystem waiting for the right tools. Give it the precise biochemical support it evolved to use, and it will relentlessly fight for your survival. Thank you for joining us on this deep dive. We'll see you next time.